I was asked to give a talk on the habitability of modern habitability of Mars. So I've expanded that a little bit to talk about the habitability of Mars in general and life on Mars and what we've learned from the recent missions and what I personally think we should do next. So the whole story kind of comes back to what Mars was like in its early history and when life evolved on Earth. So basically, this is early Mars. It's not what Mars looks like today, but in 3.5 to 4 billion years ago, we think that Mars was covered by at least a polar ocean, and there was abundant liquid water. And this was about the time that life was evolving on Earth. And the environments were similar enough that if life evolved on Earth, the question has been posed, if life evolved on Earth in these conditions, why wouldn't it have happened on Mars? The environments were being disturbed by heavy bombardment by large meteorites. But over time, Mars cooled faster than Earth. And the reason it did is because it's 1/10 the mass of the Earth. So the Earth, as time went on, developed plate tectonics. There was subduction of the crust, and the surface remained warm. Whereas on Mars, there wasn't plate tectonics developed. And the crust did not get recycled. And so eventually, the components of the atmosphere which actually react with liquid water ended up becoming part of the crust, really. So Mars quickly cooled, and conditions became much more like the present. So if that whole hypothesis is true, what might life on Mars have looked like, early life? This is a picture, which I actually took a couple of years ago, of a place that's considered the prototype environment for what Mars life might look like. It's Shark's Bay in Western Australia, and there are things that look like coral heads that are actually microbial structures. They don't have any larger life forms. Corals is actually a much more complex ecosystem, but these are formed from microbes, from basically cyanobacteria. And they form lumps that are called stromatolites, and they are formed from the trapping and binding of sediments. So why is it that we think that this is what early life on Mars looked like? Well, this is another place in the same area of Australia. It's called the Pilbara. And we can find structures that look just like these structures in Shark's Bay that are 3.5 billion years old. And these are actually the earliest evidence of life on Earth. And again, this is a picture I took. And this is somebody, standing in front of one of these outcrops of what was stromatolites. But the thing is that they kind of look just like the rest of the rocks around there. And you have to have a really sharp eye. And this is actually one of the rocks that is a stromatolite, and it has these little layered structures. So this idea has been kicking around. It actually, to my knowledge, originally originated with Chris McKay, and it's been kicking around for a number of years and has caught on. Sometimes it takes a long time for these ideas to catch on in science. But the Curiosity rover is essentially investigating that hypothesis that there's evidence of early life on Mars associated with the habitability of the planet, and that there should be structures like the stromatolites if you can find them. So the Curiosity, Jim Bell, and I'm sure probably Steve Squires gave very good talks about that. Jim Bell's talk yesterday was excellent, so I'm not going to dwell on this a lot. But just to say that the Curiosity payload includes instruments to determine the mineralogy of the rocks and to analyze for organic compounds. This is the Chemin instrument does the mineralogy. The sample analysis on Mars does the organic compounds. And so Curiosity has been driving around for about a year now. And one of its first important discoveries was this conglomerate rock. And again, this was discussed by Jim Bell yesterday. But conglomerate rock is only formed in a stream-type flow. So this is the first really compelling evidence that there was not just liquid water, but rushing liquid water. I mean, liquid water rushing over the surface. And it's the first rover-based compelling evidence for that. Curiosity has drilled in only one rock so far. And it's drilled only 10 centimeters. That's not very far. But what you see is that the surface material is red or orange everywhere, but you get down just beneath the surface and you get to this gray material. And gray actually, just by itself, implies that the material is unoxidized. It's reduced. And in fact, this is the results of the first drilling from the sample analysis on Mars. This is the organic analyzer. And shockingly, to some people's minds, no organics were detected in this reduced material. But admittedly, there are some problems with the sensitivity of the instrument. As a matter of fact, the instrument is leaking organic compounds because it had actually ampules of organic compounds that were supposed to be used to extract organics in the first stage of releasing the organics so that they could be detected by the mass spectrometer. And so it's detecting its own contamination. So that is actually reducing its sensitivity. But even within the sensitivity that it has, if there were just meteoritic infall as the only source of organics, and the meteoritic infall was not being somehow destroyed, you should see organics in that sample, and you don't. And that's a puzzle. Another thing that can be seen in this set of results, and this is just a plot of what's released as a function of temperature and detected by the mass spectrometer. What you see is that there's this compound, perchlorate, that's found on Mars, and we didn't know that until the Phoenix mission, and I'll get into that later, but this perchlorate isn't just at the surface, it's actually found all the way down into the drill core. What was also discovered was that there were clays, 20% about of clays in this gray material, and that there were oxidized and reduced sulfur and iron compounds present simultaneously. So this is a partially oxidized material. Okay, so this is, again, I'm just kind of breezing over the curiosity results because they were covered in a lot of detail yesterday. I'm going to spend more time on results from the 2008 Phoenix mission because this mission was looking for modern habitability. So again, we've got an ancient habitability story and modern habitability story. So the question is Mars was ever habitable? Is it still habitable? So Phoenix was addressing that question. And we found that it may still be habitable in some locations. It's not everywhere. But where ground ice is present, it appears to still be habitable. So the Phoenix mission landed in the Martian Arctic in 2008. And again, a mission objective was to search for a habitable environment. So what does that mean? A habitable environment, the way I'm defining it, means that terrestrial microbes, if put there, could survive and grow. Three factors are important for that: the availability of liquid water, the availability of a biological energy source, and that the environment doesn't have something in it that's nasty that will kill you. So non-toxic conditions. So some of the evidence that Phoenix found, and again, to cut to the chase, because I did this analysis myself, Phoenix found a habitable environment, a modern habitable environment. One of the first things that happened when we dug into the near surface, and again, this is kind of like the drill, you know, you scrape away the top, and what do you find? Well, you find ice, but you don't find it everywhere. It's what's called segregated ice. And segregated ice forms by freeze-thaw processes. The ice actually has to be mobile. It has to move around to form lenses of ice. So Ice wedges are one kind of segregated ice. We find them in the Arctic very commonly, and they happen when the ground in the winter, as it gets colder, contracts and cracks open, and then in the spring or in warmer conditions, liquid forms and flows into the crack, and then when it freezes again, it widens the crack, and so these things grow. Okay, the instrument carried on the Phoenix mission that actually made one of the most important discoveries is the wet chemistry laboratory. This basically took in samples and did a liquid process. So it looked at the soil with ion-selective electrodes. It looked for ions that would be potentially important for life. So a major surprise that was found by doing this analysis is that almost all the chlorine on Mars, and the surface of Mars is actually rich in chlorine, almost all the chlorine was in this form of perchlorate and magnesium perchlorate. So this is a highly oxidized compound, and it was found at like 1% concentration. Here's the paper in Science in 2009 that announced it. One of the things that's important about having this, and we think of it as a salt. One of the important things about it, having this salt at this concentration, is that the salt is extremely hygroscopic. It pulls water into the salt crystal, and its eutectic solution is liquid at -60 centigrade, which is the actual average temperature of the surface in this location. And it's actually the temperature of the subsurface. So this has actual important implications for both the habitability and probably the fact that we see evidence of water mobilization. Another thing that was discovered by Phoenix was that there was calcite mineral detected. Calcite is what forms when carbon dioxide, the main component of the atmosphere, reacts with water. And it was formed at 3 to 5% of the concentration of its whole soil mass. And the perchlorate also was found to be distributed heterogeneously. So there was essentially a hyperspectral imager on the spacecraft, and it actually had an absorption band for perchlorate in it. Not that we knew that was going to happen. It just happens that perchlorate had an absorption band in the spectral range that this imager could detect. And it was found that there was a patchy concentration of perchlorate, again, suggesting that there was mobility. Things were being moved around. Okay, so that's the evidence of liquid water at that site. The next thing is a biologically available energy source. So there's two possibilities. You either have to have solar energy or you have to have chemical energy. But solar energy is dangerous on Mars because there's a huge amount of ultraviolet. In fact, experiments by Andy Sugar have shown that you cook and kill all microbes in about a day exposed at the surface of Mars just by the ultraviolet light. So if you're going to use sunlight, you have to get under something that's transparent that will filter out the ultraviolet but leave the photoactive wavelengths, the ones that you can use for photosynthesis. So this is a microscopic image from the microscopic imager on Phoenix, and what we find is that there are some semi-transparent particles, exactly the kind of particles that would allow microbes to live basically in the near subsurface, and we find in many, many environments on Earth that have high sunlight and high UV and high dehydration, we find cryptoendolithic microbes that live just under the surface of the rock with transparent or semi-transparent grains shielding them from the hostile conditions. So that's one possibility. But the other possibility is actually the perchlorate. Turns out the perchlorate is chicken soup for microbes. There's a lot of microbes that can live on perchlorate. So this is a plausible energy source for subsurface life. Now, it isn't perchlorate alone. You actually have to use essentially there needs to be a reductant. So the perchlorate is the oxidant, and you need a reductant. So these are a list of possible reductants. And in my view, the most likely is iron, reduced iron, because we see reduced iron all over the place on Mars. So it looks like we have most of the ingredients. But the fact of the matter is, right now, it's not warm enough for metabolism to occur if metabolism is anything like we understand it on Earth. However, it turns out that the polar regions of Mars are in an especially cold time. The obliquity of Mars varies up to 45 degrees. And so we're currently at 25 degrees. 5 million years ago, we were at 45 degrees. And at high obliquity, solar flux in the northern polar region is 2.5 times stronger than it is now. So how does that translate into climate? So this is a plot of the insolation on Mars as a function of time, going back in time over the last 10 million years. And here is where we are right now. And what you can see is that most of the time, or a lot of the time, it's actually more sunlight. There are actually times where there's less sunlight. So this high-resolution component is actually the precession of the location of the location on Mars in terms of season at which perihelion occurs. It turns out that the orbit of Mars is sufficiently oblate that, or oblique, I'm not sure what the right astronomical term for this is, but basically when perihelion occurs, Mars is closer to the sun. When aphelion occurs, it's further away. And right now, northern summer, it's the summer in the North Polar Cap, where Phoenix landed, it occurs at aphelion. And that turns out on a 50,000 year time scale, that will occur, or that will repeat. 25,000 years ago, that landing site, the northern polar region, is at the perihelion position, which means the insolation is higher. And it's higher by enough that there's liquid water able to form a few days a year. Pure liquid water, not salty liquid water. But if you go back over these obliquity cycles, what you find is that even 1 million years ago, and I think this is the highest obliquity case, but as you go back in time, you get liquid water being able to form at the surface more and more days a year. This is a climate model, and you can't see these numbers, but basically the colors are telling you, and there are contours of constant temperature here, or constant number of days a year that liquid water forms. So this is now, and this is at 45 degrees obliquity, and what this is showing is that at 45 degrees obliquity, the polar regions are actually the warm place on Mars, and the equator is cold, or colder. And this is like 100 days a year that liquid water can form, pure liquid water, at the Phoenix landing site. So findings for a habitable environment are, there's evidence for liquid water, number of lines of evidence. There's a biologically available energy source, both sunlight and perchlorate, which could be metabolized subsurface, so you don't have to be at the surface. And the environmental conditions are currently too cold for growth to occur, but the other conditions would allow growth, and the climate is often much warmer than it is today. So, so then the question is, how do we search for life? And we've kind of been dancing around that question. So either past or present, how would we search for life? Well, there's a group of us that are working on this, and we propose, and this is actually coming from the work of Alfonso de Villa at Ames, that we should be searching for biochemistry, the chemistry of living systems. Because biochemistry can be used to detect both extant and extinct life. or ancient life and modern life. And that the biochemistry carries fundamental information about life, about its genetic, its physiological, its phylogenetic makeup. And there are common principles that should govern Earth and Mars biochemistry because they should both be based on liquid water. So these principles are that life self-separates from the abiotic world with lipid membranes. Life synthesizes polymers of 3 basic monomers, amino acids, nucleobases, and sugars. And the selection of monomers to form biopolymers is not random. So a group that I collaborate with in Madrid has been developing for more than a decade now an instrument called the Signs of Life Detector. The PI of this instrument is a scientist named Victor Paro. He's at the Center for Astrobiology in Madrid. And the solid instrument is designed to search for life based on its biochemistry. So how the instrument works is you put in a sample of soil or ground rock. You add a solution of salts to it. You shake it up, and then you expose it to what is called a immunoassay microarray. Immunoassay is a technology that all of you have experienced if you've ever had a blood test or if you've ever had a urine analysis. Immunoassay is used to do a huge amount of things in biomedical field. Because basically what it involves is that you can make a antibody to virtually any biological compound, biologically produced compound. And you make that antibody, believe it or not, in the skin of a rabbit. You basically introduce the compound, and the rabbit's own immune system makes the antibody to that compound. So you can print a suite of antibodies onto a microchip, and then when you expose that microchip to your sample, if you pick the right antibodies, it happened to be in the sample, they will bind with the antigen will bind with the antibody on the chip. And then what will happen is you can illuminate that. You can actually tag it with a fluorescent tracer. You can illuminate that with laser, and that cell will light up. And that's how this instrument works. So basically, this is a data product from the instrument, and this shows where the lights have lit up on the basis of a particular sample analysis. Here's a paper from Astrobiology in 2011 that describes the instrument, and here's a picture of the instrument used in the field. And full disclosure, I'm a collaborator on this instrument and potentially plan to be working with the instrument as a team member in the future. So this instrument has been successfully tested in Mars analog environments. It has detected biomarkers in the most life-depleted environments on Earth, where other life detection approaches have failed. This is a picture of a detection basically done using University Valley permafrost. University Valley is the coldest, driest environment on Earth. It's in Antarctica. And samples from University Valley were run through this instrument solid, and we're able to detect microbes where other types of technologies have failed. It has proven sensitivities down to 1 to 2 parts per billion for peptides and proteins, and it's TRL 6. That's technology readiness level 6. It means something probably to some people in this room. But it means the instrument is, it's not a flight-proven instrument, but it's an instrument that can be taken to flight readiness in a straightforward path. So, what about the next mission? There is a mission plan now using the same rover design as Curiosity. It's planned for 2020, and in something like a month, an announcement of opportunity is about to be released for instruments. So the question then is, will Curiosity, or the follow-on Curiosity, will it search for life? Well, SOLID was actually solicited as a human exploration mission contribution to the 2020 rover for information. A request for information was received on this. And it is discussed in a science definition team report that was issued that's supposed to define the objectives of the upcoming mission. However, the instrument in the SDT report was first described, and then it was described why the instrument would not fit on the mission. And it specifically stated, the biomarker detection system would be a high-cost payload with significant impacts to rover design and low crossover with the science objectives of the 2020 rover, which focuses on ancient biomarkers. Well, that was disappointing. But I think the points, they may have misunderstood the instrument. They didn't have a lot of information. They were asked for information, they were provided information, but it wasn't like a full proposal. So what are the facts? First of all, they claimed there was low crossover with the objectives of the mission because the objectives of the mission were ancient life signatures, not modern life signatures. Well, in fact, both can be detected with the same kind of instrument. The same technology can detect biological signatures that are stored in the fossil record and those which are that disappear quickly or are not very robust to long-term storage. The claim was made that it was high cost, but in fact it's contributed by Spain. There is some NASA contribution that was proposed, but it was at a pretty low level. About 1/5 the cost of the payload that was actually selected by HEOMD to be flown. It was stated that it requires special sample handling that would add complexity to the surface operation scenarios. But in fact, the mission plans to drill. In fact, the whole mission is about collecting drill cores for sample return. And when you drill, you automatically produce cuttings. You can't not do it. And cuttings is what the instrument actually can use directly. And we have already ideas and simple mechanisms to capture and deliver cuttings produced while drilling. It was also stated that it was not mature, but it's actually at TRL 6. So now why do I know all this? Well, it's because we've been working on a mission to detect life, modern life, at the Phoenix landing site for a number of years. This is a mission called Icebreaker. And the idea is to return to the Phoenix landing site with a drill to get to the subsurface. Well, why would we want to drill? So turns out that galactic cosmic rays will kill organisms basically by destroying their DNA. Or whatever their, assuming it's something like DNA on Mars, whatever their replication mechanism is, basically because organic compounds are just broken down by galactic cosmic rays over time if there isn't metabolism to repair it. So this is actually from a paper by Dartnell in Geophysical Research Letters in 2007. This is a plot of the dose received as a function of depth for this is three different kinds of models. So pure ice is the dotted line, and dry soil is the solid line, and a mixture is the one in between. So basically, and this is the kill time as a function of depth. And this is for the most robust organism there is on Earth, radiodurans. So basically, at the surface, you got about 18,000 years. If you haven't metabolized and somehow repaired yourself in 18,000 years, you're toast. But as you go deeper, the time gets longer. And when you're down a couple of meters, it's up to hundreds of thousands of years. So you've got a pretty good chance to make it through or a time when, again, as at the Phoenix landing site, there are times when it's extremely cold and dry and there's no metabolism possible, but other times when it's actually quite pleasant and there's lots of opportunities for metabolism. So for that reason, the Icebreaker mission is based on drilling. And this is the Icebreaker drill. It's built by Honeybee Robotics. It's been tested in the Antarctica. It's been field tested in the Antarctic twice. Just came back from the field this last austral summer. And it's a very high TRL drill. We think this drill also is TRL 6 and could be flown on the Icebreaker mission. So in conclusion, we think that subsurface sampling is needed to reach modern life that's still alive. Biomarker detection of ancient life may also require access to the subsurface, since GCR, galactic cosmic rays, will also degrade organics over time. And maybe that's why we're not seeing organics in that near-surface material that Curiosity has drilled. So in summary, or in conclusion, Sarah Palin had it right. Drill, baby, drill. In the front? Sorry? The Icebreaker drill was really designed to go 1 meter. There are other drill technologies that I've worked on that have gone as deep as 10 meters. I personally think that more needs to be done to understand this galactic cosmic ray, to the interaction of the galactic cosmic rays with the potential habitable environment in the near-surface ice. And it's something that I have kind of overlooked, and I just discovered it a couple of days ago when I was preparing this talk, that there really is a case that you might have to go down, you know, as much as 5 meters. Let's see. Let's try you, sir, and white shirt.
Yes, I was wondering, is a solid instrument being considered for the European mission to Mars?
Yeah, that's an interesting story. The ExoMars mission actually solicited proposals, but it was many years ago. And there is another type of instrument technology that was out there in the European community. And as a matter of fact, the Spanish team that built this instrument did, in fact, propose to ExoMars. They did not win that competition. There was another instrument that did, and that instrument, for whatever reason, I actually don't know what the reasons are. That instrument has been descoped from ExoMars, and I don't, so it's no longer on the mission.
Oh, yeah, the red dragon, how deep is that supposed to go? Would that have your instruments on?
So that's this is a gentleman who's been following the news. I've been working with a team that has been exploring using the SpaceX Dragon as a Mars lander. And we've done actually several studies on that. And our first study was actually exploring taking the Icebreaker payload, the exact Icebreaker payload, which includes the Signs of Life detector instrument, and a deeper drill, and going to the Phoenix landing site. So that, yes, that's actually a really good way to get deep, because the Dragon's actually very tall. You can get a 2-meter drill inside it without even having to have replacement strings. You know, just one big tall drill. And it's actually string change-out that's one of the most risky elements of doing a drilling mission. Let's see. Hold on.
Someday, humans were able to terraform Mars and give it a breathable atmosphere. In the absence of a magnetosphere and plate tectonics, would Mars be able to keep such an atmosphere? And if not, what kind of replenishment would be necessary? Are there any estimates of that from what we know today about the surface and current atmosphere of Mars?
Well, what you're referring to is actually one of the big mysteries, you know, that in order for there to be a warm, wet Mars ever, you know, early conditions in which there was a liquid ocean, for example, you have to have a much thicker atmosphere than you presently have. And that is always assumed to be a carbon dioxide atmosphere at something like 5 bars pressure. So this is 5 times the pressure in this room. it is a mystery. It's a big mystery that large quantities of calcite, you know, white cliffs of Dover, are not seen everywhere on Mars. They're just not seen. So it's never really been fully explained where is this missing calcite, because there should be a lot of it. So if I understand your question specifically is, could you initiate plate tectonics on Mars? I think that's geophysics. No, this is ring world engineering.
In the absence of plate tectonics, would Mars be able to keep an atmosphere if by artificial means in the future we could give it one again?
I see what you're saying. It would not be easy. So especially if that atmosphere were carbon dioxide. Because the problem is that carbon dioxide reacts with liquid water and it forms calcite, which is why we have such huge calcite deposits on the Earth. You know, the carbonate rocks, which are everywhere, are ubiquitous on the Earth, and that's why we don't have a huge greenhouse in our atmosphere. I mean, that is what's giving us only a few hundred parts per million of carbon dioxide in our own atmosphere. The rest of it is tied up in the rocks. So that process actually happens very fast. You know, the water reacting with carbon dioxide happens very, very fast.
Hi.
Sorry, somebody's...
Hi. I just wondered if there's been any evidence from any of the rovers for caves on Mars.
Yeah, so yes, there is evidence for caves on Mars. There's karst topography, actually, but of course, you know, there aren't caves you can crawl into. No one's crawled into them, let's put it that way. But there are skylights that have been observed that probably are the type of thing that is involved with lava tubes. I mean, they're probably lava tubes, I think. But there are holes in the ground that you can't see down into, and they have been observed by the HiRISE imaging instrument. Excuse me, over here?
Have you taken a microscope into the field, into Antarctica? And then the second part of the question, could that be automated for Mars in order to screen a large number of samples in order to select a subset of those samples, say for a sample return? How many samples in Antarctica are you screening in order to find samples that might be valuable, say as an Earth analog to bring? How many samples does it take? Are there problems with the type of sample that if you would screen enough, you'd be able to select a subset of those samples that would be worthwhile bringing back? And then I guess there must be something with the microscope that is technically difficult also.
Well, microscopy is, I'm not sure what you're referring to. Microscopy is a really lousy way to look for biology. I mean, it's just not, it's impossible to separate without a lot of sample handling to separate what is biology from what isn't with a microscope. And you have to be looking basically in thin section at extremely high power and, or, you know, scanning electron microscopy perhaps. Now people are trying to develop scanning electron microscopy for space flight. But it's, again, you know, it's a difficult problem because the higher and higher resolution you go, the less you know about the context. How are we detecting the fluorescence in this instrument? Or how about we talk about this offline? I think you may be misunderstanding how the instrument works. The fluorescence is detected in the instrument with a CCD camera. So you just take a picture of the chip.
Wait.
We'll talk later.
Okay.
So I don't know who's had their hand up longest. Okay, you.
Hi. In our search for life throughout the universe, I sometimes wonder if our focus is a little bit too narrow. I just wondered if your group ever kind of thinks outside the box and maybe even speculates and looks for other life forms that don't exactly fit our template?
Yeah, the problem is, I mean, I agree with you. The most compelling search for life, the most compelling sort of, I guess, result you could get in searching for life would be if you actually found a separate genesis of life, or a type of biochemistry, or a type of life form that was completely alien or completely different than the type of chemistry and life form that we're based on. And that's, a lot of people think about that. You know, there's the whole field of astrobiology, but the problem is it's hard to think about it because it's like, how do you know what to look for if you don't know anything? You know, it's, how do you know what you don't know? So instead, I think the approach, a better approach, is to look at environments that are plausible and to try to understand those environments better. So the environments that we think are the most likely to host Earth-like life, Earth-like meaning biochemically similar to Earth life, is the environments of Enceladus, which is a moon of Saturn, and the surface of Mars, and possibly the subsurface of Europa. The type of environment that you might look for a life that would be completely biochemically different than Earth would be the surface of Titan, because, you know, all the environmental conditions are very, very different. So that's kind of the best one can do, I think.
Can I follow up on that? In your talk, you identified a disequilibrium environment on Mars. You know, classically, people have said that if you could find a disequilibrium environment like methane and oxygen in an atmosphere, that was a signature of life. Now, on Mars, you say you have metallic iron and mag chloride, chlorite, whatever. So, have you considered a metabolic cycle of organisms that could take advantage of that disequilibrium and of life forms that might use it?
No, but it's an interesting idea. You, sir.
Assuming we eventually find active microbial life, what are the potential interactions between human colonists on Mars?
The potential interactions. So, you know, the first thing to understand is there a potential interaction? The reason that the human exploration part of NASA was potentially interested in this instrument is because they have a, what do they call, a strategic knowledge gap. So they have a roadmap that has identified certain topics to be important to understand before humans are landed. And one of those topics is there life on Mars, and is it potentially hazardous to humans? So they want to know, is it hazardous? So that really is the question, right? Is it hazardous? So if it's hazardous, then the set of interactions is going to be different than if it isn't.
Last question.
Tony, I'm getting Tony. Yes.
Carol, if you have to go as deep as 5 meters into the permafrost to find something that's not destroyed by galactic cosmic radiation, does that permafrost ever melt at the temperatures you're talking about over time? I mean, I'm just wondering, is there a dichotomy there? You have to drill so far that it would be below where something could be alive or it lived.
Well, again, I think that we need to understand better the interaction between essentially there's a modeling study I have in my mind that, unfortunately, I'm not a modeler, so if I can get a good modeler to team with me on this, that actually models both this radiation environment and sort of when conditions become habitable and how long it takes to double and grow during those times, to see, you know, kind of where that balance point is. I think it's probably not as bad as it's 5 meters under the ground, but without having done that study, and just having looked at this guy's paper and said, whoa, you know, what he's saying is it's 650,000 years is the kill time down to 3, you know, at 3 meters depth, and it's less time as you go higher than that. I think, you know, the modeling study needs to be done to see what is the kind of the optimal depth to look for life.